Hello and welcome to Biomentors. Today, our topic of discussion would be on androgenesis. In the last lecture, we discussed about gynogenesis. Both androgenesis and gynogenesis are type of haploid culture. And this lecture would be helpful for the students pursuing their BSc and MSc courses in botany or any life science subjects. So, what is androgenesis? So before looking at the androgenesis, we have already discussed about haploid culture, but still we are going through the introductory portion, that what is haploid plants. Haploid production of plants through anther or microspore culture is called as androgenesis. Like in the last video, we discussed that haploid production of plant is called as haploid culture. In when the part of female part of the plant is used, then it is termed as gynogenesis. And when the male part of a flower is used, then it is called as androgenesis. Generally, the two main part of the flower are used in androgenesis. Either anther or microspore, which means pollens, are used in androgenesis. This, as it is a haploid culture, so they are haploid in nature and N. Most commonly, the androgenesis is seen in species of Solanaceae family and the Poaceae family. And the examples of this are tomato for Solanaceae and Poaceae for rice. In androgenesis, immature pollen grains are induced to follow sporophytic mode of development by various physical and chemical stimulations. So in this method, immature pollens are induced to produce plantlets under various physical and chemical stimulations. Types of androgenesis. So as it is discussed, if we take anthers in the culture media for the culturing, then it is called as anther culture. And if we take microspore, or we can tell at this is pollen grains onto a culture media, then it is called as microspore or pollen culture. Now, the first we will discuss about anther culture. So the technique of anther culture was discovered by Ghova and Maheshwari in 1964. So the first step of anther culture is collection of the explant. So the collections of anther is collected. The success of androgenesis is generally seen when anthers are collected from a plant during its flowering season. So mainly we have to take care of the flowering season of the plant. Usually in the flowering season, the flowers are collected. Why? Because during the flowering season only, the male or the female part of a plant is reproductively active. Usually, anther at the stage of flower buds will give better response during androgenesis. So at the early stage, it will give a better response, right? Pre-treatments to the anther. As we know the anther structure, uh, if you have studied the type of the anther wall development, then inside the anther there is a chemical which is called as sporopollenin, and it is resistant to stress and mechanical stimulations and chemicals. So for the breaking of that anther wall, we need a pre-treatment. Once the donor material containing the microspore has been selected, it requires a specific treatment. The treatment can be applied in different phases of explant, such for intact flowers like barley. Whole spike is pre-treated. So in barley, the whole spike is pre-treated by isolating anther. So the second method is by isolating the anther itself when they are pre-treated, or by isolating microspores. If we are culturing only the microspore of the plant, then microspore are pre-treated. So what are the types of pre-treatments? The first pre-treatment is cold water pre-treatment. In this, the temperature is 33 to 6 degrees Celsius for 15 days is given to such type of flower or anther, right? And the result of cold treatment is given for a week or 15 days. For non-viable anthers and microspores are killed at it will regard aging of the anther, right? After that, we have to check the viability of the pollen. So with the type of chemical, they will also check the viability of the pollen before culturing to get high percentage of success in culture. Hot water treatment, generally 30 degrees Celsius for 24 hours. The plantlets are kept for 24 hours. For 24 hours at 30 degrees Celsius, and this is hot water treatment. And for xerophytic plants, 40 degrees Celsius or even high temperature is given to break the sporopollen or to check the viability of the anthers, right? Now, step 2 is in the step 2, flower birds are now sterilized. Now, we have collected the explant. Now, we have to sterilize the explant. So, where we have to sterilize the explant? They are sterilized in laminar airflow chamber. Young flower birds with immature flower bird are surface sterilized and rinsed with sterilized water and sterilized distilled water or deionized water. So as we have seen in gynogenesis, after collection, the flower was surface sterilized. Same way in this process also the flowers are surface sterilized. The callus from the flower bird will be removed. The calyx, sorry, not calyx, callus, calyx. The flower is a portion which is called as calyx. Then after corolla, and then after the reproductive parts of the flower is there. So by mistake, this is callus is written. We have to remove the calyx, and then after the corolla is separated, and then the anther part of the flower, we can see the anther. And gently they are separated with the help of filament, with the help of scalpel and forceps, we will remove the anther filaments to avoid the injury to the anther, since it induces callus formation for the other anther walls. So they are gently removed from the flowers. Now step 3. Each anther is gently separated and put onto the nutrient media. So each anther is taken onto the nutrient media and they're separated. So this was the step. Now we will see in the figure what is there. So first we are collecting the flower in step 1. Then they are surface sterilized. Then after they are cultured on a nutrient medium. And then finally it induces embryoid formation, haploid plantlet is formed, or callus is formed, and then after haploid plantlets are formed. So here also we can see two types of anther culture, either direct anther culture directly from the embryoids or anther, haploid plants are formed, or indirect, which means callus is formed in this type of anther culture, and then after haploid plantlets are obtained. So these were the types of anther culture and the main steps for anther culture. Now we will look at the types of anther culture. So on the basis of uh, which we have discussed, that there are two types of anther culture, direct and indirect. Now we will see this culture in detail. So in direct anther culture, anther, a globular heart-shaped anther or the embryo is formed directly, and in indirect androgenesis, grains instead of normal embryogenesis divides differentially into callus. So callus formation is seen in indirect type of callus culture. And generally, indirect type of callus culture is seen here. The examples are given, seen in rice, wheat, and tomatoes. Now, pathway for the formation of the embryo of the plantlets can be of 4 different types. So the first is uninucleate pollen grain divides into unequal daughter cells. So 2 unequal daughter cells are formed. Both of this undergoes division and it will contribute into sporophytic development. In the pathway 1, unequal division of the cell is seen. In second pathway, in this uninucleate pollen grain or the anther provides an equally, so it divides equally, which results in formation of a vegetative and a generative cells. The sporophyte of the plant that rise to form for division in the vegetative cells, and the generative cell does not divide. So vegetative cell divides and generative cell does not divide in path 2. In pathway 3, in the sporophyte of the plant rises due to the formation of sporophytic cells. In this part, vegetative cells are not divided, only the sporophytic cells are divided. So now we will look up the figure. In the first pathway, unequal division is seen. In the second pathway, the generative cells, in the second pathway, vegetative cell divides and the generative cell doesn't divide. And in this third pathway, generative cell divides and vegetative cells doesn't divide. Now we will look at the fourth pathway. In the fourth pathway, both of the cells are dividing, vegetative as well as sporophytic. Both the cells divide in this type of pathway. The pathway 1 differs from pathway 2. So in pathway 1 also both the cells were dividing. In pathway 2 also both the cells were dividing. But what is the major difference? The major difference is that in pathway 1 and pathway 2 differentiates the pathway 1 differs from pathway 4 in this differentiation of the cell, which means in the pathway 4 differentiation of cell is seen, while in pathway 1 differentiation of cells is not seen. So as we have seen in pathway 1, there is unequal distribution of cells, while in pathway 2 there is equal distribution of the cells. So in this figure, pathway 1, 2, 3 and 4 are labeled. There is a mistake in this, but my explanation in this slide is correct, but by mistake I got a different figure, I think. Right. So please refer this portion of the figure only, not this portion. Now we will look at the microspore culture, which is culture of pollen grains. So in this also, the pollen grains are taken and then after they are kept on a nutrient media, but it has a slight different procedure than the anther culture because the size of the pollen grains are very minute. In the gynogenesis, there was no different method because ovary and ovule. Ovary is also bigger in size compared to the anther, while the ovule is also bigger in size compared to the pollen. But pollen grains are very minute in size, and thus there is a different method for the culture of pollen grains. Step. First, the collection and sterilization. So first, the flowers are collected and they are sterilized. The flowers are generally collected in the bud stage. The same procedure which is we have seen in anther culture. Step, collection of pollen and maceration. So now the step differs. So we have to collect the pollens from the flower and then we have to macerate it. With the help of forceps, anthers are removed. Anther are collected. Microspores are squeezed out of the anther by pressing them against the side, and with the help of a glass rod. With the glass rod, we have to break that and we have to pull all the anther out from the pull out or take out the pollen grains out from the anther to macerate it. We can also maybe in this process different chemicals can be used. Step 3 is filtration. So the debris are removed by filtrating the suspension through a filter paper or a nylon cloth. So then after we have to filter with a filter paper or nylon cloth. Now step 4 is centrifugation. The suspension is then centrifuged. When the suspension is centrifuged, the supernatant containing the debris is discarded. So when we centrifuge, there are two portions in this test tube, which is obtained at the end. At the end, the upper portion is called as supernatant, and the lower portion is called a pallet. So generally, the pallet is used. Not the supernatant is never used. Generally, in the centrifugation process, this is seen. So pallet is the supernatant is discarded, and the pallet is taken. Pallet of the pollen is resuspended in fresh media and washed at least 2 to 3 times. So then this pallet is washed at least 2 to 3 times. And then after we have to do the step 5, which is isolation of pollen and keeping them on a nutrient media. And finally, we will take the pallet and we will wash that with the different distilled water or the different solution. Then after we will put this pollen grains onto a nutrient media carefully with the help of a pipette. Okay, we can take the final suspension culture and then we have to gently put them onto a petri dish or a test tube. Incubation. Petri plate is then filled with paraffin, paraffin to avoid dehydration. So paraffin is not there, paraffin is there. It is a liquid, a yellowish tincture, yellowish liquid, which avoids dehydration, and it is incubated for 28 degrees Celsius, and they are incubated for at least 14 days. After 14 days, the culture is transferred onto a different medium. The microspore from the embryo on the callus, which may be later differentiated into the haploid plantlets. Then after we will get haploid plantlets from the pollen grains. And finally, the hardening procedure, we will shift those plant in greenhouses. So here is the figure or a diagram showing the haploid culture. So first we will take the the plant's flowering portion, and we will remove the anther. Then we will sterilize that explant. Then we will macerate the pollen grains from the anther. Finally, we will filter that solution. Filter that solution. Then after we will centrifuge. We will take microspore, which is a supernatant, a pallet in this case, and we will discard the supernatant. And then finally, we'll put this microspores onto a nutrient media. And this was all about microspore culture. Now we will look at the factors affecting pollen culture. So the first factor which affects the pollen culture, or we can tell both the type of culture, which is anther culture and pollen culture, both, the condition of the floral bud. So the time of natural flowering conditions like daylight, humidity is the best to collect such. At that time only the flowers are collected, and generally the young flowers are collected. Any infection or stress to the donor plant will lead to less success or complete failure for induction of androgenesis. So if the selected plant has a stress or they have an infection, then it will not lead into formation of suitable plantlets. Right. Age of the donor plant is an important factor. As I mentioned, the age of the flower should be young enough. Anther derived from an older inflorescence shows decline in frequency of the haploid production due to reduction pollen viability. They are less viable. The second is that genotype of a plant is affecting the androgenesis. The genotype of the donor plant plays a major role in determining the success and the failure of gynogenesis. Some genotype of some species may show androgenesis and some may not show the androgenesis. So this was the all about genotype. The third is growth inhibitors. Anther wall sometimes develops the anther wall. Sometimes the development of microspore inside the anther wall may be interrupted due to the formation of certain growth inhibitors. Okay. Substance leaking out of the anther wall in contents to the nutrient media during anther culture. And this way, a possibility is that this type of growth inhibitor will affect the formation of plantlets. And so it is very much important to give pre-treatment to an anther culture to remove the inhibitors, right? So now the fourth point is that physical factors. So what are the physical factors? Generally, the main physical factor is temperature and humidity. So physical factors such as temperature, light, and humidity are maintained. Generally, the temperature used is 23 degrees Celsius to 28 degrees Celsius for anther culture or pollen cultures. It is observed that chilling of anther before isolation increases the number of pollen grains. But this also depends on the type of species, whether we have to chill the flowers or not. Okay. On the other hand, it is observed that high temperature of the anther when given to result brassica before transferring them to the normal temperature results in formation of embryoids. So this is also an example where high temperature induces the formation of embryoids. So the temperature factor completely depends on the type of plant used in an anther culture. So mainly the temperature is playing an important portion in formation of anther. Okay, so sorry, in formation of plantlets or embryoids. Other factors such as organic supplements can be added to the media, such as coconut milk. For example, coconut milk, vitamin, nucleic acids, and amino acids should be added to the media. The last factor is culture media. Type of culture media which is used mainly plays a key role in induction of anther culture or pollen culture. So the culture media also plays an important role. The media should contain the correct amount of growth regulators and inorganic nutrients. The growth regulators mainly used in such type of culture is auxin, which promotes the induction or the division of pollen callus. Kinetin or cytokinin is an essential for induction of pollen embryos in solanaceae. So this is an example in which chitin induces the formation of embryos in Solanaceae family. The concentration of sugar also plays an important role in induction of pollen haploids. Generally, activated charcoal is added to the culture media. Why the activated charcoal is added to the culture media is that there are two reasons. There are two reasons because it removes the inhibitor from the agar media, and as it removes the inhibitor, it will induce the growth of the induce the growth of the callus culture and then after results in formation of pollen culture. So this was about factors affecting the pollen culture. Now we will look at the advantages of the pollen culture. Now, overcrowding of pollen grains in an anther is estimated, and isolation of pollen grains are equally exposed to the nutrient media. Unwanted growth of the anther wall and other associated tissues are eliminated. So, if there are certain inhibitors present in the anther wall, with the help of pre-treatments, these inhibitors are removed, and thus formation of embryos, and then after finally the plantlets can be formed. Right? Steps of androgenesis can be observed starting from single cell. So it can be taken from a single cell. Various factors governing the androgenesis can better be regulated. right? Pollen is ideal for uptake, transportation, and mutagenesis study. So pollen culture is very helpful in mutagenic studies. As we discussed, it only contains androgenesis or gynogenesis, only contains a single parent chromosome. So we can study the mutations which are caused by recessive parents. Pollen may be directly transferred to the embryoids so that it is very suitable for understanding biochemistry and physiology of the androgenesis. So this is the advantage. Other advantage, higher yield of haploid plants per anther could be expected in pollen culture than the anther culture. So there is a higher yield of a plant for anther culture could be expected in pollen culture. So expectation ratio or the success ratio of pollen culture is higher than anther culture, which is mentioned here. And I don't think this is an advantage of anther and pollen culture. This is a point. If it is asked that which culture is most suitable, pollen or anther, then we can tell that anther culture is more suitable. So what are the disadvantages? Now we will look at the disadvantages of the anther culture. So the plant may originate not only from the pollen grains but also from various parts of anther. So this is a disadvantage of anther culture. During anther culture, there is always possible that the somatic cells of the anther that are developed will also respond to the culture conditions and not this is not a soap, and reduces unwanted diploid plantlets. Sorry. So formation of unwanted plantlets can also be induced in anther or pollen culture. Sometimes a cellular division may not take place because often fails to grow in vitro. In vitro condition, sorry, this is again a mistake, sorry. In vitro condition, it will fail due to lack of nutrition. Anther or pollen culture will fail to develop. And this would result into abnormalities and cannot grow. The main disadvantages of haploid culture is that it is very expensive. And this is the last advantage. With this, we end our lecture. If you like this video, do give this video a big thumbs up and do subscribe to our channel for more videos. And in the next video, we will discuss about cell suspension culture and protoplast culture. Thanks for watching.